Hi, I am Rahul, and you're watching Scientified Human. Let's talk about part 2 of cellular respiration, that is Krebs cycle. We learned about glycolysis, which is an anaerobic process, meaning it does not require oxygen to occur. As oxygen is not necessary, the primitive, or first simple organisms on Earth were able to generate energy through glycolysis for millions of years. But 2 ATPs for a glucose molecule just is not that much. In order for higher organisms, like animals, to evolve that can run, jump and swim, they had to evolve additional metabolic pathways that generate more energy than glycolysis. This became possible once plants covered the Earth, thereby filling atmosphere with oxygen, which is the end product of photosynthesis. Thus, to generate more energy, the possibility for large-scale oxygen-dependent aerobic respiration was born. The process occurs in the mitochondria of a eukaryotic cell. If we look at the endosymbiotic theory, mitochondria seem to have been entirely separate organisms that were incorporated into eukaryotes specifically for the respiratory abilities they possessed. This activity starts with the pyruvate molecules that are generated in the cytoplasm during glycolysis. This pyruvate molecule will enter the mitochondrial matrix to combine with coenzyme A. In the presence of NAD+, pyruvate will undergo decarboxylation. Oxidation by NAD+, and then attachment to coenzyme A, generating acetyl coenzyme A. Acetyl coenzyme A then enters a cyclical pathway called tricarboxylic acid cycle, more commonly called Krebs cycle, after the scientist Hans Krebs, who first elucidated it. This is a stepwise pathway requiring different enzymes. The tricarboxylic acid cycle starts with the condensation of acetyl group with oxaloacetate and water to yield citrate. Here the enzyme, citrate synthase, removes acetyl group and tacks it onto oxaloacetate to form citrate. Next, with the help from aconitase, a water molecule is removed, and another one added to form a structural isomer of citrate, called isocitrate. Then catalyzed by isocitrate dehydrogenase, isocitrate is oxidized by NAD+, and then decarboxylates to form alpha-ketoglutaric acid. Next, another carbon dioxide is removed, and further oxidation by NAD+ takes place, with the help of alpha-ketoglutarate dehydrogenase. The resulting molecule will join with coenzyme A, once again to form succinyl coenzyme A. Coenzyme A is then displaced by a phosphate group to form succinate, which is catalyzed by succinyl coenzyme, a synthetase. This will make one molecule of guanosine triphosphate, or GTP, in the process, which can be used to make ATP. Then, with the help of succinate dehydrogenase, succinate is oxidized by a different molecule, FAD+, which will result in fumarate and FADH2. Next, fumase will catalyze hydration, which will result in malate, and lastly, one more oxidation by NAD+ takes place. With the help of malate dehydrogenase, to give oxaloacetate, which restart the cycle by reacting with new acetyl coenzyme A. Overall, for every acetyl coenzyme A that enters the cycle, will produce 3 NADH2, 1 FADH2, and 1 ATP. Since one glucose molecule form 2 pyruvates in glycolysis and therefore 2 acetyl coenzymes, we can double these numbers per glucose molecule. We can see that there still has not been a huge payoff in terms of energy. But the products of tricarboxylic acid cycle will then move to oxidative phosphorylation, which will generate the majority of the ATP produced in aerobic respiration by the metabolic pathway called electron transport system, which we will discuss in the part 3 of cellular respiration. Thanks for watching, guys. If it helps, then don't forget to hit the like button, comments down below, share and subscribe to my channel for more tutorials.